Hello students, this is Gina Piscatelli giving a lecture on some basic principles of neuron function. This lecture will deal with just the resting membrane potential and how that changes in the neuron when it's stimulated. It deals with study question number 7. So let's just review some structure of the neuron first. Over here we have dendrites labeled A, and then we have the soma, and B is labeling a part of the soma called the axon hillock, where it tapers before the axon right here, which is C. And then the axon terminals, of course, are out in the periphery here, labeled D. This is true for a basic motor neuron, so we'll look at that in a minute here. So since our whole purpose is to talk about how neurons function, let's first just discuss what a nerve impulse is. A nerve impulse appears to be the flow of positive ions or positive charge down the axon. But it turns out that actually a nerve impulse occurs when, and I want to show you this slideshow. Let me just see if I can do it. Yep. What really happens is that there's a series of inward and outward flow of positive ions or cations. So this particular picture shows just a region of the axon. Here is the box, and so this is magnified greatly, and I'm going to play that animation again. But what you're going to see is that cations, instead of starting at one end and moving down the length of the axon, what they really do is leave, and then others come in, and then those leave and others come in, and then they leave, and so on and so forth. So let's do that animation one more time, if it'll let me. Come on. Here we go. In, and then that goes out, and another comes in. So each move of ions in and then out is called an action potential. And an action potential triggers a neighboring action potential, and so on and so forth. So there's a series of individual action potentials that occur down the length of the axon, and we call that a nerve impulse. So in order for us to understand how that nerve impulse can happen, we've got a number of things that we need to talk about, and this lecture will just be 2 of those. One is, what is the electrical condition of the neuron at rest when it's not conducting a nerve impulse? And then part 2 is, well, how does that get disrupted or change so that we can start a nerve impulse? And then in another lecture we'll talk about an actual action potential and how it gets propagated. So some basic principles that we need to understand in order to talk about neuron function is, what is current, what is voltage, what is potential, what do these things mean, and why are we going to use them? Let's start really basic. You're familiar with the word current. It's a flow of some kind of electrical charge. Like in your house you have wires, and there is charge flowing in those wires. So that's current. Voltage is actually a measure of a force, which is due to the attraction of opposite charges. So the negative charge would be attracted to the positive and the positive is attracted to the negative. By how much they're attracted is a measurement that we can take with a voltmeter. We call this voltage. And the potential is very similar to that. It is the difference between voltage at 2 points. They use it to mean the potential to do work. Because if you have a force moving in a certain direction, there's the potential to do work. And since we are talking about cells, we always talk about a potential to do work across a membrane. And so we're going to look at the difference in charge or voltage across a cell membrane. What's the charge in the cytoplasm and what's the charge outside? And so that we know what the force is. And we call that a membrane potential, which we'll come back to again. So the membrane potential of a neuron at rest is called the resting membrane potential. And it is said to be polarized, this membrane, because there is a positive charge outside and a negative charge inside the cytoplasm. So the difference is 70 millivolts. So this is a cross-section of part of the neuron, we'll say the axon. And notice that this is the cytoplasm here where it's negative, but the extracellular fluid is positive. So that's a potential, and the neuron is currently at rest, and the difference in charge is 70 millivolts. Now the reason we use the negative sign is because we just always refer to the cytoplasm, like what is happening inside the neuron. So this is not exactly how, it's not very detailed, but this is how that has been measured. There's something called a voltmeter, and here's a neuron at rest, and they can do this with the aid of a microscope. They can put a little electrode in the cytoplasm, and then they can have a grounding electrode in the fluid outside of the cell, and this voltmeter measures the difference in charge. So this information can also be graphed. If you look at a neuron and what it's doing at rest versus when it's conducting a nerve impulse, you see that at rest it's at negative 70 millivolts. But then when it begins to conduct a nerve impulse, that changes, and the inside of the cell becomes more positive, but of course it's going to return and it's going to go back to rest. It overshoots a little bit, which we'll talk about later, but it eventually gets back to the same value. So what causes this resting membrane potential? Well, inside the cell there are negatively charged molecules like proteins, DNA, and in addition there's sodium-potassium pumps in all of our cells, neurons included, that pump more positive ions out than they pump positive in. So remember, 3 sodium go out, but only 2 potassium come in. So that's a net positive on the outside. In addition, the membrane is selectively permeable, meaning it's permeable more to some things than others. It is not permeable to sodium. Notice that there is a concentration gradient for sodium. If it could, it would diffuse inward. But it can't because the membrane is not really permeable to sodium. Then if we look at potassium, potassium has a chemical gradient where if it could, it would it would diffuse outside of the cell because it's higher inside than outside due to the sodium-potassium pump. However, the membrane and the membrane would allow that too through some what we call leaky potassium channels, but the potassium is attracted to the anions, so it tends to not leak very much. That's what keeps sodium high on the outside and potassium high on the inside, and contributes to our resting membrane potential. So the forces that I just talked to you about can be described in two different ways or categories. One category is chemical gradients of ion concentrations, and the other are electrical gradients. So chemical gradient, there's an inward force for sodium. There is an outward force for potassium. However, you know that the sodium is blocked. It can't get, I can't draw with this thing. Okay, it can't get into the cell. Electrical gradients refer to just charge. Both sodium and potassium are attracted to the anions, and so both would stay close to the anions if they could. Sodium can't get in there though. So the first thing I'm going to have you do is draw a circle on a piece of paper. So I drew my perfect circle. And what I want you to do is make sure you know the distribution, meaning the amounts, and where sodium ions, potassium ions, and anions are across the cell membrane. So remember, sodium is really high on the outside, potassium is high on the inside, and then there's also some negative anions inside. And what establishes this is the sodium, potassium pump. Okay, that didn't work. That's potassium pump. The cursor doesn't keep up with how fast I go. Sorry. Okay, then I want you to think about, why doesn't sodium diffuse down its concentration gradient? And then what force drives potassium out of the cell, chemical or electrical, if it could go, and why doesn't it go? That's number 5. And what is the value, the numeric value of a resting membrane? So I have the answers here for you. The reason that sodium doesn't diffuse into the cell is because the membrane is not permeable to sodium at rest. And potassium could diffuse out of the cell according to its chemical gradient, but then why doesn't it? Because of the electrical gradient keeping potassium attracted to the anions. And the value of a cell's resting membrane potential is, oops, that's supposed to be a 7, is negative 70 millivolts. So now we're going to look at part 2 and what disrupts the resting membrane potential. So it's stimulation. A neuron has to physically be, well, chemically be stimulated in some way. And what that does is it changes the resting membrane potential. And when that change occurs, I guess the change itself we call a local potential. So it's a localized change in membrane potential. And it usually occurs at a dendrite. There's a dendrite. And the stimulus that causes the change from negative 70 to something else could be due to a chemical, pressure, heat, or light, like in your retina. And what the stimulus does is it changes the membrane permeability to those ions we talked about by opening or closing a channel. And therefore it changes from negative 70 to something else, which we call a local potential. Sometimes stimuli are excitatory and sometimes they're inhibitory, but we have to talk about what that means before that makes sense to you. So let's talk about a neuron first being excited. Okay, an excitatory stimulus happens of some sort. Let's say a chemical binds to a sodium channel, which is shown down here. This is in the neuron. When the stimulus or the chemical binds to the sodium channel opens and the sodium will go into the neuron. Then the inside of the neuron becomes more positive than -70. Why does it become more positive? Because sodium is positive. So if we look at our little graph, here's resting membrane potential. A channel was opened, sodium went in, and now the inside of the cell is more positive. We now say that the membrane is depolarized. It's not polar anymore because the inside and the outside of the neuron charge, anyway, becomes more similar. So that's what happens with an excitatory stimulus. An inhibitory stimulus will be the opposite. But what ion is affected is an ion that we haven't talked about yet, and that's chloride ion. So normally chloride is high on the outside of the cell. So that's my little cell. And chloride ion is really high on the outside. So the membrane starts out at rest at -70 millivolts, and that's shown right here. And then a chemical of some kind, here's a chemical, binds to an ion channel, and that ion channel is specifically for chloride. So we call it a chloride ion channel. And chloride will then move in to the neuron. And now the inside of the neuron becomes even more negative, and you see that drop right here. Why does it become more negative? Because chloride ion is negatively charged. We then say that the neuron is hyperpolarized because the inside and the outside are even more different than a difference of 70. Now notice that it's like only -75, but it's still more polarized. So these local potentials that we've just talked about are due to some sort of a stimulus. And stimuli vary. Sometimes they last a long time, sometimes they're strong. So we say that these local potentials are graded. They vary. They vary in how much the membrane potential is changed. And that really just relates to how long is the ion channel open. If the sodium ion channel is open for a long time, that's a strong stimulus. If it's not open for very long, then it's not a very strong stimulus. What's most likely, though, is a neuron is going to receive several stimuli at once or over a short period of time. And those stimuli are summed up. We use the fancy word summated. But it's just because it's a cumulative effect of all the ions that come in. Now this local potential is reversible in the sense that it doesn't necessarily lead to a nerve impulse. It depends on how strong it was. Okay, so in order for a nerve impulse to actually occur, that local potential has to reach -55 millivolts. And then an action potential will happen and you can't stop it. So the action potential is irreversible. That's supposed to say irreversible. Okay. So in your textbook they use some fancy words called EPSPs and IPSPs. And I'm just going to tell you what they stand for. They're really not that necessary. They're just examples of local potentials. E stands for excitatory, I stands for inhibitory. And the reason that there's these other letters here has to do with how they ever discovered that neurons change their resting membrane potential. So I'm going to show you this picture right here. underneath that line. So let's say a presynaptic neuron right here, this first one, okay, is conducting an impulse down to its axon terminals. It forms a connection or a synapse with another neuron. In this postsynaptic neuron, they measured what is the effect. What is the effect on this particular neuron when there's an impulse that comes here? And they found that sometimes it's excitatory and sometimes it's inhibitory, depending on what chemical was released there. That's way too much information. But excitatory EPSPs are excitatory, and the PSP stands for postsynaptic potential because of where they measured it. They measured it in a postsynaptic cell. This cell, the first presynaptic cell, obviously had to have a change in membrane potential, resting membrane potential also. But an IPSP is inhibitory and an EPSP is excitatory. That's pretty much all you really have to know. So this is a picture that gives you an idea of how multiple stimuli can change the resting membrane potential. So in this particular picture, all of the blue axon terminals are releasing or conducting excitatory stimuli, and the red ones are all giving an inhibitory stimulus. I haven't actually counted them out, but let's say that your first excitatory stimulus would change the resting membrane potential just a little bit, up to 69. The second one a little bit higher, up to 68. But those two are added together if they happen close enough in time. So you would actually get something like this. But then an inhibitory stimulus comes along very soon, and that makes the membrane potential go towards negative. If you sum all of these together, you get something kind of in the middle. This is what we mean by graded. It's different depending on how many different stimuli and how frequently they come together, what the effect is on the neuron. So some things that you want to get out of what I just talked about is, are number 7, does a stimulus cause a change in resting membrane potential throughout the entire neuron or just locally? And of course the answer is locally. I have that written on a separate slide. When an excitatory stimulus acts on a neuron, will the inside of the neuron become more positive or more negative? And the answer is more negative. And then here, number 9, binding on of an inhibitory stimulus would cause what ion channel to open? And that's chloride, because it's inhibitory, and that's the only ion that we're ever going to see that's inhibitory. And it'll diffuse into the neuron. And what does depolarization mean? Depolarization refers to the fact that there's a change in the polarity. Initially, the cell was positive on the outside and negative on the inside, and that's going to change. So the next slide lists all the answers for you, and I'm going to post this. Thank you.